So, first of all, let me just say I'm delighted to be here. This was, a phenomenal invitation to come talk to this community that I've sort of watched grow over the past few years. And so I'm really honored to be here. Let me just sort of give you a little overview of what I hope to talk about. So the title of the talk's a little long, but I promise we'll explain it all by the end. I'm going to talk about sort of a vision for a way to view workflow systems like Nextflow, and what we might be able to do with them that we're not currently doing, right? Things that aren't really that much, hopefully, of a stretch to imagine doing, but that would be supported by the model that we currently have. So I'm Rob Patro. I'm an associate professor in the Department of Computer Science at the University of Maryland, and also affiliated with several other, institutes, including the Center for Bioinformatics and Computational Biology. So, full disclosure, I'm coming at this from the bioinformatics angle. It switched up here, but it did not switch here. So, sorry.
It's gone black.
Cool. All right. Thank you. All right. Would not be a conference without some AV issues. All right. So I want to just sort of do the acknowledgements and conflicts up front. So first of all, the acknowledgements. One of the main things I want to talk about today is the idea of automated provenance tracking. And this is work that was really spearheaded by my collaborator Mike Love and I. We have a grant where this is one of the main topics. And then I've listed here all the co-authors on the main paper that describes the scheme that we've come up with, which is for a tool called TXI Meta. I want to thank seqs Labs for inviting me to give this talk, and also to our funders, including specifically NIH, NSF, and CZI. And CZI has been mentioned several times already, and again, it's sort of one of these unique opportunities for people who really care deeply about software and developing and maintaining software to get funding for that. And they've funded us over several rounds of their different programs. And finally, I'll mention the conflicts. I'm a co-founder and CTO of a company called Ocean Genomics, which does some genomics in the cloud. That's not primarily what we do, but I wanted to declare that up front. So, also some caveats up front. This is my first invited in-person talk since COVID began. I've given some webinars, but excuse me if I'm a bit rusty, and not in a good way. Those of you who know me know I'm an advocate for the Rust language. This is not the kind of Rust I'm talking about. This is also my first Keynote talk, so we're going to go on this adventure together, and forevermore this will be the first time I've ever given a Keynote. And it's also not really what I'm used to talking about, right? So I'm used to talking about recent research from our group. I pitched some ideas. One was to talk about some of the integration that the Nextflow core team has been doing of Alvin-Fry, which is our single-cell processing tool, into the single-cell RNA-seq NF core pipeline. But ultimately, we decided to go a different route. And when I pitched this talk idea to Phil, it seemed that people liked it. And I literally said, OK, YOLO, we'll give a completely different talk on a different topic than I normally give. So I want to start out motivationally, right? Both from a positive perspective, but then also just sort of asking what we might achieve and what we might want to achieve. So if I think back to myself, say, a decade ago, right? Rob is, a graduate student, actually at that point, just starting graduate school, so let's say maybe like 7 or 8 years ago. And he's doing some, genomics, bioinformatics, and stringing together shell scripts, basically. And we think about what would the bioinformatics workflow dream be, right? What would we really want to be able to do in sort of an automated fashion? What would we want our workflows to support? What kind of capabilities would we want to have? And so I've listed some here. I'm going to go through them one by one, right? I mean, the first one is sort of really the minimum viable product, right? You want a workflow to be able to be to produce a reproducible analysis, right? You want to be able to have the same inputs, get the same outputs. You'd like to be able to control versions of software. Virtualization was very early at that point, but you'd like to be able to virtualize dependencies and things like that. You want workflows to be scalable, right? So you'd want to be able to run your workflow on one sample or many samples for an experiment for a paper, and then if it's a really nice tool, you'd like to be able to do this at scale, right? Maybe on thousands of samples. You'd like modular and reusable components. You know, you don't want to have to rewrite the same thing over and over and over again. This is a general principle of programming languages, but I think it's also a principle of workflows. You'd like the slick interface, right? That's sort of a bonus. But we're going for what would be the dream here, not necessarily what would be the MVP. You'd want uniform tool interoperability. This is one that I'll talk about in detail. And you'd want this provenance and metadata tracking, and I'll argue why. So let's take a quick tour through these different desiderata. The first thing I mentioned is reproducible workflows, right? You want the same input, the same output. You want controlled versions, virtualized dependencies, et cetera. And I would argue we're there, right? I mean, there are, you know, always some very, very strange edge cases depending on deploying on different platforms and things like that. But for the most part, this works brilliantly, right? And so the picture I have here is of a run of an NF core single cell RNA-seq pipeline. And you can see it sort of lists here the tools that are being run, relevant paths, the different profiles that are being used, the input sample sheet, all of the output information, some specific information about parameters, the protocols that are being used. This is displayed to the user, but of course it's also tracked in the system, right? So you have this information. This makes reproducibility literally a button push. So success. Another is scalable workflows, right? So you'd like to be able to take the same workflow and run it on a sample or run it on hundreds or thousands of samples and not have to think very hard about what you have to do differently, right? The workflow system is supposed to figure out things like scheduling and dependencies for you. And I would argue we are certainly there, right? So these images I have here, the one on the left is a tweet from Nick Wong. Many of you may follow him on Twitter. He frequently posts these runs from NF Tower, where you can sort of see 19,000 jobs pending, 13,000 jobs completed, 150 currently submitted, and 50 running. So, you know, this is scale. And then the example on the right is from a largely Nextflow-based tool for processing SARS-CoV-2 data. The corresponding paper is published in Genome Biology for this Climb COVID project. And the x-axis is date of things processed, and the y-axis is cumulative CPU hours that were spent on doing it. And you know, the number of jobs here accumulates to 123 years of CPU time managed by the workflow system. So success number 2, I would say astounding success. We want modular and reusable components, right? So there was some talk about this already. We don't want to have to rewrite the same thing again and again, right? If I write a sort of small module that takes a BAM file and sorts it by coordinate or something like that, I don't want to have to do that separately in every workflow. I'd like to have a component that does this, like a function in a programming language, and I can share that function. And maybe I can group functions together like a library, and I can share that library. We are building that, right? That is being built. So here, for example, the example I came up with is NF core modules, right? Currently, when I took this, it's probably different now. When I took the screenshot, it listed 632 different modules. And I did use the term here sub-workflows. And I have to mention, I swear that I wrote this slide before I learned they actually existed, right? But I actually think that this is sort of a natural extension and a very good one of the idea. We have small little bite-sized units that are modules. We have slightly larger ones that are sub-workflows. that naturally compose into workflows, right? Okay, so astounding success number 3. And then I would be remiss if I didn't mention the desiderata that wasn't part of the minimum viable product, but something that's really nice to have, right? So when Nextflow Tower first launched, and I tried it out, it was just one of these moments where it was sort of transformational for our ability to track our experiments and things like that, right? And to get that feedback interactively. I remember it was like 2 o'clock in the morning. I started off a run for experiments for one of our papers. I pointed my grad student at the URL, and we just watched the jobs run, right? So really, really cool. Astounding success. I forget where we're on now, number 4, right? Okay, so now let me get to the meat of what I want to talk about. There are some places where I think we could do more, potentially. And so one of them is uniform tool interoperability. This is a very challenging problem, right? If you've worked with bioinformatics tools, you know that there's many different input and output formats. There are different kinds of dependencies, there are different versions, and even when there is a specification for a file format, something like SAM or BAM, you may have noticed that not all tools follow it precisely, all right? So there's some difficulties there in really hooking things together in a transparent way, and I'll give a concrete example later on. And so I'd argue maybe we do have a little bit of this. So this image here is from a merge pull request that does a conversion of a particular format of single cell count data, MTX, to another, H5AD, as a conversion module, right? So clearly this is the kind of thing that modules can handle, but maybe there are other ways to do this, and maybe there's ways to scale this up better, right? So this happens currently mostly at the edges of workflows, and it's something that we have to be explicit about. And then here's one where I think we could do a lot better, which is automated provenance tracking. So provenance and metadata of the data that we're processing is crucially important. I'll give a couple of examples why. And we like to manage and propagate it automatically, right? It's not exactly the kind of thing that we are good at doing manually, right? That's much better than doing it by hand. And currently, you know, we can do something towards this. If we track all of the input and we have file names and things like that automatically tracked, that's much better than what would happen if we said, okay, we ran a bash script, and I don't really remember exactly what version of GenCode I used or whatever. And in that case, the information's lost. Here, there's a file associated with it, so I could probably do a much better job of sort of figuring out the inverse problem of exactly what input I used, but maybe those files got moved or deleted or whatever. So, you know, what are these files? Where do they come from? Were they renamed at some point? Will this limited information that I have about the input even be propagated to the output, right? It might exist in sort of a log, but what I'd really like is for provenance information to exist where the output of my analysis does, so that anything that uses it downstream has all that information for me. Okay, so the next question that I want to ask is, I've given at least 2 places where maybe we haven't had astounding success yet, but I think we could. And how do we get there? How do we get closer to the ideal? So this is where the maybe unparsable terminology in my talk title came from, because it's something that I made up. All right, so I made up this term of augmented execution context, or AEC. So if you don't know what it is, don't feel bad, because I don't think it's a thing yet. But I wanted to try to take the ideas that I had about what I'm thinking about and put them into a definition that we can work with. So I'm going to define an augmented execution context as something that's present when the process that's coordinating a computational procedure or a workflow has complete information of all of the steps that the procedure is going to do and control over how they're executed, right? So, I mean, this is not a new thing, right? I would argue that workflow languages usually have this. This is just a new way of naming it, right? But I think that names are important, right? And they guide our thinking. And so I think naming it in this way might unlock some potential capabilities that we haven't been thinking about. So I'd argue that Nextflow workflows are usually augmented execution contexts already, and the same is true with snake make recipes and Airflow workflows and things like this, right? Not all workflows are. Some of them are very agnostic to the execution environment, but I don't think that's necessarily the case for most of them. So the claim that I want to make, the thing I hope to convince you of, is that augmented execution contexts provide the type of environment we need to support these desiderata that we're currently missing, right? The ones that I think haven't really been achieved yet, as well as potentially many more that people, that are creative and energetic can come up with. So let me talk about these in reverse order, right? Because I'm going to spend actually more time on provenance tracking than I'm going to spend on interoperability. And I want to talk about it by way of example. And the reason for this is it's not something that I think we have uniformly in the community yet, but we've built a tool to help do it in a very particular use case. So the motivation I'm going to use here is a tool that we built called TXI Meta. The origin of the name is its transcript abundance importing with metadata. It builds off of sort of some prior work led by Charlotte Sonneson for this tool called TX import. Okay, so if you don't do RNA-seq analysis, you might not be familiar with these tools, but the general idea is that one of the first steps in an RNA-seq analysis pipeline is to quantify the reads and figure out the abundance of every gene or transcript in your sample. And once you've done that, you usually want to load your quantification information into a statistical package like R, and then do an analysis, figure out which genes are differentially expressed with a tool like DE-seq or EdgeR or Limavoom. There are many of them. And TX import is really this sort of layer that sits between, and it says, okay, you could have used these 4 or 5 different programs to do your quantification, and you might want to export to these 4 or 5 different tools that do differential expression. Here's one package that makes loading that information and converting it easy. And what TXI Meta adds to that is this automated provenance tracking, okay? So that's what I want to focus on. And if folks are interested in more detail, the paper that describes TXI Meta was published in 2020 in PLOS Comp Bio. So part of the motivation for this is when we were sort of originally thinking of these ideas, Mike and I had a set of problems that we ran into on a regular basis, right? So, we often do analyses for other folks, Mike more so than myself. And, sometimes questions would come up like, what particular reference annotation do you use? And the answer was not always obvious. And so Mike posted this Twitter poll. When working with collaborator or public summary data, how much of a problem is it to obtain a genome or gene versions for the data that you have? And so the answer here is, surprisingly, only 10% of the respondents said it's not a problem. Now, I don't claim that Twitter polls are scientific. There's definitely a sampling bias here, but this is a really strong signal, right? 45% of the people said hassle, this is a hassle, can cause a mistake, and 45% of the people said, sometimes I can't even determine what particular genome or annotation version was used. And as a follow-up to this, he asked the question, do you think that software to automate reproducibility of these kinds of analyses is a good thing? And again, the answer was an overwhelming yes. Not 100%. There are some folks who thought, well, if you can't solve the problem perfectly, then you shouldn't try to solve the problem. But this was a strong enough signal to see that we weren't alone here, right? So far so good. Let's talk about a typical RNA-seq analysis and sort of what it might look like at the first step. Okay, so when you're doing RNA-seq quantification, you have sort of 2 main pieces of information that the process starts out with. You have some reference transcriptome, okay, so this could be, if you're working in human, an annotated version of the human transcriptome from GenCode or Ensemble or UCSC. It could be, if you're working in a non-model organism, a de novo assembly, right? It's just the set of things that you're going to quantify, the gene transcripts you think could be present in your sample. And then on the other side, you have the data, the raw data, right? FASTQ files in this particular instance. And a typical pipeline will build an index over the reference transcriptome that can be used for every analysis for that organism and reference version, and then it will quantify the reads, right? And the output, in this case, if you use our tool Salmon, is a set of files, each one called quant.sf, that gives you the abundance information for all of the transcripts in that sample, right? So if you have 3 input samples, you have 3 output quantification files. And this part's pretty straightforward, right? And this part here, the reference transcriptome, contains all of the sequence of the reference transcripts. That's the part I want you to remember. That's what we are quantifying. Okay, so later on, you have this quantification information. And if you don't know exactly all the analyses you want to do beforehand and have set them up, this might be just sitting on disk somewhere, right, in some folder. And so then you have questions you might want to ask later, like, are the genes that are differentially expressed enriched for ChIP-seq peaks, right? Or are they enriched for certain methylation signals, right, in relevant conditions? And this might work out beautifully and brilliantly if we have been very meticulous in tracking all of the information, but it might not. What if we don't quite remember exactly what version of the genome we used to generate the quantification information? Or we don't remember exactly which genes we quantified, right? Transcript versions change constantly. Even in well-annotated organisms like human, I mean, we all know that, there's this recent T-to-T assembly, which has added new genes and transcripts to the genome. So we're still updating even the most highly studied organisms' annotations, right? So these things are still changing actively. And so if you don't know which ones you used, then you have this sort of very difficult process in front of you of trying to figure it out, to piece together the information, right? So here, we're performing this bioinformatics analysis where we want to check for enrichment of ChIP-seq peaks with differentially expressed genes from quantification information. And so here, let's say, okay, well, we know maybe not the precise version, but we're going to use this transcriptome database from HG38, known genes, to get the set of transcripts that we're interested in both quantifying and doing enrichment on. We have to go through this process from our RNA-seq abundance estimates to convert to gene names from, between UCSC and Ensemble. We want to take the intersection of all the transcript IDs because, there might be some differences in what's annotated here. And we get a set of genomic ranges. The red box here is potentially a warning that we would want to report to the user. Some things might be dropped from the analysis, right? In the conversion between the different databases and in the places where there are differences in intersection of annotation, we might lose some things. Okay, so this is a lot of work, right? We have to do a lot of bookkeeping to get where we want to be. And it can even be worse than this. Maybe we don't even have really any idea where the quantification information came from. So we it's human, but we're going to ignore the specific genome version, and we're just going to hope that we have some overlapping identifiers. Hopefully people wouldn't actually do this in practice, but you'd be surprised or maybe not what you see. And so in this case, I don't know exactly what version, but here's a file I found in a related directory called genes.bed. There's a bed file that probably has my gene ranges. And then what happens here? Well, it can be really bad, right? Like, I mean, maybe you didn't find enrichment when enrichment actually existed because your annotations didn't match up and you missed some important parts of the signal, right? So my argument here is that there's a lot of work that goes into getting this kind of analysis right, and a lot of it requires deep scientific thinking, right? Trying to understand mechanisms for why things might happen. But a lot of it doesn't, right? A lot of it is something that is sort of monotonous work that is much better left to a computer than to a person. So this sort of leads to this idea of forensic bioinformatics, which is if I get that information out and I don't know all the information I want to know about it, I have some questions that I have to ask, right? So there's like a role here for a forensic data scientist. I have a set of features that I've quantified. What are these features, right? How do we know that they were correctly labeled in the first place, right? Like how do we know that they're from a trustworthy annotation? Who generated the data, right? What resource is it from? What's the provenance of it? Is it Ensemble? Is it UCSC? Something like that. what versions of the software were used to generate the data, right? We talked a little bit about how this can be done in the context of Nextflow. So, this is something that can be tracked. And so you have this problem oftentimes in reality. Maybe not quite this bad, some of the boxes are filled in, but you have to figure out what to put in each one of these boxes when you don't know the precise version. And this is not a good use of an analyst's time. And it's also not something that as people we are generally great at. And so you'd like a few things to happen, right? You'd like, when you have this quantification information, to not have missing metadata about the transcriptome provenance. You'd like to be able to do an auditable analysis, right? And by that I mean computational reproducibility from the raw data. But for that you need to know what the raw data is. And the reads are only part of the raw data, right? The annotation is another part of the raw data. You'd also sort of like direct access to the annotation, right? In general, I'd like to load up my information in R if I'm doing an interactive analysis, and I don't want to have to think hard about what transcript database do I have to pull in, what annotations do I have to pull down. I'd like to have that kind of thing happen automatically. I'd like to be able to do analysis at different levels of resolution, right? So a lot of the annotations we have for genes, at least, exist at various levels of resolution. There's transcript level and gene level, all the transcripts sharing a particular transcription start site, et cetera. A good annotation has that. I don't want to have to deal with that manually. I'd like all that information to be populated for me. And I'd like things to be placed in genomic context, right? So ideally, if I know the version of all the annotations that relate to each other, I don't have to pull in 3 different annotations for different types of data. I can put them all into one genomic context, which allows me to easily do things like overlap chip peaks with regulatory regions and genes that might be DE. Okay, so how do we get there? I'm going to talk about the very specific solution we came up with for propagating transcript annotation metadata and provenance. And so the idea is we're going to use hashing, okay? And there's sort of a common saying that there's only a few tricks in computer science. Hashing is one of them, right? Indirection is another one. So here's the basic idea. We have a setup very much like we had before, except now our FASTA database is with the reference transcriptome is colored purple. And here's why. It has some source, okay? And the source in this case is GenCode v38. That's the annotation we're using. So what we're going to do is when we build this index, which is the index our quantification tool is going to use to quantify this RNA-seq data, we're going to hash the reference transcriptome. There are many specific ways to do this, but what we're going to do is we're going to go through all of the sequences. We're going to make a manifest of them as a hash value, and we're going to store that hash value with the index. It's not going to be part of the index. And now every sample we quantify is going to be quantified against an index. That index has a hash value that points to the specific reference that's used, and we're going to propagate that hash information to all of the output, right? So one thing that's changed here is the output is no longer individual files, quant.sf, they're entire quantification directories, right? And the reason for that is there's more information in them. Now every directory has a JSON file that says, this is the specific provenance of the annotation I quantified against. Okay, so this is the way the system works. There are many ways to solve this problem. For now, just imagine we're using a good hash, something like SHA-256 of the cDNA sequence. And then at analysis time, you're able to do something like this, right? So this is actual R code from TXI meta. I have a sample sheet, which is what call data is. I load a experiment object, and it gives me this nice output. It says I'm importing the quantifications, I'm reading in the files that are TSV format. I found a matching transcriptome. It's GenCode Homo sapiens version 38. I'm loading the transcript database. I'm generating genomic ranges in R for all of the transcripts, and I'm fetching the genome information. And the output is a summarized experiment, right, which is a R object in the bioconductor ecosystem. And the user didn't have to do anything extra here except use a version of the quantification tool that supports this information. Okay, some details about this. This works out of the box for GenCode, Ensemble, RefSeq, common organisms, common model organisms. There's some other engineering stuff, right? So you wouldn't really want to have to reproduce pulling down the annotations and creating the genomic ranges for every analysis you do if you're doing them on the same system. So it makes use of BioC file cache to only download and parse the GTF once per system. It makes use of Annotation Hub to deal with pre-parsed GTF databases. You get information that is then stored locally and cached, like the transcript to gene mapping, transcript to exon mapping, all of the relevant BioC objects if you're working in that ecosystem. One thing that Mike found when working on this is that what RefSeq calls latest is not necessarily stable, though it does appear to have a permalink. And then the other thing is you can create this JSON object for sharing called a linked transcriptome, right? So all the stuff up top works great if you're in a model organism, but what if you're not, right? Like what if you have your own assembled transcriptome that you want to do this on? So here's an example of that. This is actually where some of the other co-authors were critical in figuring out how to do this for non-model organisms. And the idea we came up with here is basically if you have your own assembly, here we have a Trinity assembly that we would like to make a linked transcriptome for. You can generate the provenance information for this specific transcriptome in R, in TXI meta, using this make linked transcriptome function. And then you can upload that to a repository. And now we know what the provenance of this transcriptome is, right? So later on, we can say, okay, here's this JSON file that gives me provenance information. I could have it locally, I could have it stored in the cloud automatically, and I can load a linked transcriptome. And loading a linked transcriptome will pull down all that provenance information for the custom annotation. And so this lets us deal with things like de novo transcriptomes, diploid transcriptomes, multi-species transcriptomes, which don't have their own annotations unless you make them. And then other types of particular annotations, like maybe you include spike-ins in your reference, right? That's something that the reference might not have, or fusion genes. Okay, TXI meta is not alone. There are several people working in this space. It's actually, I think, a very interesting space. Some other examples are Verse, which does this for variants, seqs, which is a generic sequence digest, and I've listed some of those resources here, right? So there are sort of very related approaches to solving these kinds of problems, some of them more general than the ones I've mentioned here. All right, so back to Nextflow and augmented execution contexts. So TXI meta is great, but it only tackles this problem for one tool chain and pipeline. I would argue a good one, but it's one small type of analysis. What about other RNA-seq pipelines? What if I use star and then feature counts for quantification, or RSM or Callisto? And what about other analyses altogether, right? It's not always obvious what to do, and we need this for other kinds of analysis. So ideally, we'd like automated data provenance propagation to not be functionality that should have to be solved by each individual upstream tool on a tool-by-tool basis. We did this in Salmon because we wrote Salmon and I edited the code to do this, but we don't want to put that burden on every tool designer. Reference fingerprinting makes sense actually as its own analysis step because it would be easy to do, but it would also be easy for folks to forget or elide it, right? Like to not put that step in. And so augmented execution contexts like Nextflow, I think, are the ideal place to solve such an issue, right? This could be a module, it could be a part of every pipeline. We could have provenance manifests that can be used to decorate workflows in a tool agnostic way, regardless of tool by tool buy-in. So the developer of the tool would not necessarily have to buy in and create their own provenance tracking for us to keep this metadata around in the workflow. And a key aspect, I think, of this is that provenance tracking happens automatically and that the manifest lives with the analysis results, right? Because that's ultimately what you're going to be doing the analysis on at the end of the day. And you could imagine other things too. With user permission, we could create a manifest registry for custom references, host it in the cloud, pull down and push reference information on every run. Maybe you have a local repository, so every Nextflow run you've ever done has all the provenance information tracked. And we could build support for provenance of annotations and maybe even read data, right? This could be expanded, I think, beyond just what annotation you used. Okay, so that was sort of the meatiest part of the talk, because we actually have a sort of solution there. But I want to talk about one other topic too, which I think is a related place where automated execution context could be useful. So that's uniform tool interoperability. So I want to think about, for a second, augmented execution contexts as universal translators for common analyses. And I said I'd given a concrete example of this, so here it is. Single-cell RNA-seq count matrices. I've listed here several of the data formats that exist for single-cell RNA-seq count data. MTX, cell ranger format, which could be MTX, or H5, zipped, or whatever. ANN data, Loom, a Seurat object, a single-cell experiment. You get the idea. And they're all packages that support tremendous kinds of analyses with these data, but they're not transparently interoperable. So the right solution, of course, is that we should just devise a new format. Okay, so we're gonna come up with the one format to rule them all, and then we're gonna get hounded on Twitter with this XKCD comic, because of course the right solution is not to invent a new format. Okay, so we're not gonna do that. Luckily, there are several tools that do these kinds of conversions, right? The Zell converter is one. It transforms many different of these formats into other formats. Another one is SCEasy. There's one, it's part of a tool that my lab makes, in a tool called Pyro that can convert between several of the Python-based formats and MTX. And this is already sort of being worked on in practice, right? So this is an open issue in NF-core scRNA-seq as feature requests to make output compatible with NF-core scFlow, which is downstream analysis. And so this is possible because Nextflow knows how the single-cell RNA-seq output was generated, what tools were used, what format the output comes in, and what format scFlow expects, right? The interface of these workflows is known by Nextflow. And I would argue that from that perspective, the transformation can be made seamless to the end user. The person using these workflows don't even have to know that it happened. But can we make this even easier and more ubiquitous? So single-cell count matrices are just one data type. And format conversion is a much larger issue, and many conversions might be involved, more involved in this. And as tools grow, so do the number of formats produced. I mean, we're guilty of this. We built a single-cell RNA-seq analysis tool, and it outputs in a standard format, but with extra data, right? So there's a step you have to do to sort of extract that extra data from the output. And so the question here is, can we make conversions less work for developers, not just for the end user? And perhaps. So the idea I want to pitch here is, let's take a cue from programming languages. I'm a big programming language fan. It's not my research area, but I like to think of myself as an amateur PL guy. And I want to think of format conversions kind of like type conversions, okay? So maybe you've seen this before. This is a really simple example in C++. We can convert types. I have a floating point number that's 0. I can cast it to an integer. I can output it. It's 0. Here's a more interesting one in my favorite language ever, Rust. And it's meant to sort of mimic the kind of transformation we might actually care about. We have two different matrix types, MTX foo and MTX bar. They both have number of rows, number of columns, and a data element. And what I want you to pay attention to, it might be a little hard to see, is down here, we create an MTX bar and put it in a variable MB, and then we do this conversion. We get the variable MF, which is an MTX foo from MB. And so this is Rust's way of representing this idea. There's a from trait. And this from trait says how to get one kind of thing from the other. In this case, how to get an MTX foo from an MTX bar. And it can be trivial or not. Here we modify the data, right? Maybe the indices all have to be incremented by 1 in an MTX foo compared to an MTX bar. And if we run this, we get exactly what we want, right? The right number of rows and columns, and the data is appropriately transformed. All right? And so I can imagine an analog of this in a workflow system. So imagine channel conversions, right? Either explicit or implicit. This is a design decision. And here I just sort of made something up. So ignore the syntax. I just want to convey the idea. But how might this look? We might have arbitrary rules or functions that define what valid conversions are possible. They can be modular and shared between pipelines. And potentially it could even be implicit. So there's a programming language called Scala that's very sort of famous for this, implicit conversions. But the idea is, if I need something, a y, and I have an x, I can just ask for the why. And if there's an implicit conversion in scope that can provide that conversion for me, it will just sort of happen. This is the kind of thing that a workflow could manage, right? So here, our first rule, our first process creates quantification information in MTX quant format. And then we get input as ANN data from MTX in the second process, okay? So if you're allergic to the bracket notation, obviously we could come up with nicer notation. Okay, so I'm just about at time here, so I wanna close off. I think augmented execution contexts are not necessarily fundamentally new things, but a way of more fully characterizing what workflow engines like Nextflow actually give us, some capabilities that they could already provide if we just thought about them that way. I think provenance tracking and improved interoperability are just two important challenges that augmented execution contexts might help us tackle. And so sort of call to arms to the community here is, what are other ones? What are better ones? Your minds together are much more creative than mine, probably individually too. And so what are other low-hanging fruit here? We could even imagine doing really interesting things, like wild things. Augmented execution contexts could identify and address inefficiencies in a pipeline, right? Once we know, this is sort of like executor optimization kind of thing. And maybe even totally wild things, right? So like imagine something like an augmented execution context and GPT-3 give a rough draft of a human parsable method section for your paper, right? Because we know all of that information when we build the workflow. Okay, so in closing, I just want to give this message, right? The tooling and infrastructure that we have available is already well beyond what I would have dreamed a decade ago. I wouldn't have thought that we'd be here with the ability to do the things that we can. Now we're mostly limited by our imagination and creativity. And so I think it's on us to build the future of computational workflows that we dream about. All right, I'm happy to take any questions. I want to thank again my collaborators, specifically Mike Love, the co-creator of TXI Meta and author of many of the slides I used here. And you can follow me on Twitter for mostly bioinformatics and Rust-related content. Thank you.
Many thanks for that talk, Rob. That's fantastic. I think certainly I felt the pain that you describe, and I think many of our audience have as well. So you hit the spot there.
Apologies if I triggered anyone.
There's some notes of hope there, though, so it's good. We've got quite a lot of questions on Slack, so we're not going to be able to take all of them now, but maybe you can answer some on Slack afterwards. But I'll read out a few here. We've got some thumbs up. Christian asks, from a non-bioinformatics background, tracking metadata is nice, but how do you figure out what you need to track and what you do not need to track? But he's seen that sometimes people aren't brave enough to say that we don't ever need this information, let's not bother with it.
Right. So that's a great question. I'm not sure I can give a great answer other than to say that this is a meta question, right? So what information matters in an RNA-seq analysis is something that people who have done many RNA-seq analyses have learned over the course of time by trying to do different things, right? And missing certain key pieces of information. So we think we've sort of figured it out for the most common types of analyses, but I would argue even for bioinformatics analyses, it's not well known for every type of analysis exactly all the information we should be tracking. One argument here is that more is better in this case. It's always better to track more provenance information and ignore what you don't need later than to track less. But I think really we have to think hard about each specific type of analysis we want to do and first answer the question, what metadata is essential? And that's what we should end up tracking.
You touched on this a little bit at the end, but maybe you could just go into a bit more detail how you imagine this working. The hashes that are stored, so where would they be stored? Could they be stored anywhere, or do we need like a central registry, which is the source of truth?
Right, yeah, so that's a great question. So the way that it works in TXI Meta is that there is a central registry. And I think that is a good way to go about it, because then we have a place where, especially if we can allow people to upload there, we can create a larger and larger repository of known annotations. there are caveats there, right? Like, what's the access control? Do we trust anyone to upload to it? What if it goes away? What if 2 different repositories disagree about something, right? So I think that problem hasn't really been completely solved beyond the way we've solved just how we distribute annotations generally. But also, there's no reason it needs to be a completely public thing, right? You could imagine having an annotation repository locally, just within your organization, right? Or locally on your computer. So for TXI Meta, we can pull it down from online, but it need not be that way. But I do think that the integrity of that information is something that does need to be thought more deeply about.
I guess if you trust your own reference data you have locally and you can compute hashes from that.
Right. I'd be much more worried about not having it than someone maliciously changing the hash for the wrong annotation.
Coming on to some of the stuff you mentioned at the end, Priyanka asks for interoperability. What about the additional resource overhead if we're spending a lot of time translating between different data types?
Yeah. So that's an excellent point. There's the additional resource overhead in terms of time, and then also maybe in terms of space, because you can imagine some of these conversions are not lossless, and so you want to keep both formats around. Here, I've been thinking about it mostly in terms of when I build a workflow, and some rules need a particular data format that other rules don't produce, we have to solve that problem anyway. If we want to put these pieces, these tools together, we have to do that. So it's more a question of how to make that seamless and implicit rather than bulky and explicit. But if you imagine having this everywhere, I do think that sort of that optimization of how do I make sure I don't create the same thing too many times or do too much work is something worth thinking about, but I think it's a solvable problem, right? You could avoid redundant creation of things, maybe with a hashing scheme or something like that. And then, you could also imagine having resource utilization rules for conversions like we currently have for processes. Like, I want to do this conversion, but I don't want it to take more than an hour, right? And maybe the augmented execution context could even predict, okay, well, that's not going to happen. You're trying to give me a 1 terabyte BAM file. I'm not going to be able to do that.
Maybe I can just pick one more. One from Christian again. Suppose, you kind of just mentioned this, if we're doing type conversions and we lose information between those steps, so for example, from A to B and back to A again, can you think of any way we could track the error that might be introduced and kind of set some sort of threshold or limit on how much uncertainty you're willing to take?
That's an excellent question. Currently, when we do these kinds of conversions, loss can happen, and usually that's okay as long as we don't lose something that is critical for the analysis we want to do with the converted to format. If we really want to think about having just one piece of information that we can always guarantee seamless, interoperability between, then we probably want to keep some form of the original, right? And then we can go through multiple steps of lossy conversion, but we always have the original. In terms of knowing the delta of what changed, I think something like that would be great, and it would be great to have a generic system for doing that. It does seem a bit like a hard problem though, because some of these formats can be very different. Like MTX is a text-based format, HDF5 is in binary format, so there's not an easy way to take a diff between them unless we have a tool that knows what they all are, right? But you could imagine maybe something like that. Rosetta Stone that can read all the formats and then do the diffs between them and store that information. Great.
I think there's a lot of interest in working on these topics. Certainly there was some work during a hackathon on provenance tracking and things, so I know maybe we've just found our topic for the next NF Core hackathon.
Thank you very much.
Let's get started.
Thank you all for your attention.